i-CRIM Centralized Laboratory, University Kebangsaan Malaysia. i-CRIM Centralized Laboratory was officially founded in 2007. i-CRIM Centralized Laboratory provides technical and consultation services to scientific communities. In i-CRIM Centralized Laboratory, we have three main labs: Molecular Structure Characterization Lab, Physical Characterization Lab, Morphology Lab. Each lab is equipped with high-end instruments and runs with quality scientific and technical services, capacity building, competitive rates. For more information, please visit our website at www.ukm.my/i-CRIM.
Assalamualaikum and good morning. So do you guys hear me? If you can hear me clearly, please type in the chat box. Okay, thank you. So Assalamualaikum warahmatullahi wabarakatuh and good morning everyone. Thank you for joining us for the webinar on polarimeter technology and in natural products, pharmaceutical and other research applications. My name is Talia Bukade and I'm the moderator for this webinar. For everyone information, this webinar is the second webinar organized by i-CRIM Laboratory this year. The objective of this webinar is to help the participants understand the basic and applications of polarimeter analysis. In addition to creating the platform for instilling practical knowledge and networking, this webinar also aims to create awareness for the high-quality analytical services available at i-CRIM Laboratory. Upon completing this webinar, we hope participants will be able to integrate and apply the knowledge which will be helpful in improving the quality of research and publications. Before we start, I would like to go through some housekeeping rules of this webinar. Housekeeping number 1, please mute your speaker and video during the webinar. Please submit the questions in the chat box. I will read all the questions during Q&A sessions. Please fill out the feedback form before the webinar ends. Slides and recording will be emailed after the feedback form has been completed and submitted. Before the webinar ends, all participants are invited to switch on respective camera for photography session. Now, without further ado, let me introduce our adorable speaker today. Our speaker is Mr. Naraj Ravichandran. He is an application chemist in Lab Science Solutions in Berhad since October 2021. Previously, he was employed as a chemist in Encom Corp Care, a pesticide manufacturing company for 3 years, handling multiple analytical instruments such as AAS, HPLC, UPLC, Autotriterator, and also ICP-OES. Currently, he is active in providing on-site application training and analytical application solutions to customers, conduct technical seminars and workshops, assist customers to troubleshooting application problems, and assist customers in method development where applicable. Okay, Mr. Naraj, now the platform is yours.
Thank you, Miss Dalia. Can you hear my voice? Yes. All right. Okay. Uh, all right. Good morning, everyone. My name is Centralised Ravichandran, and I'm an application chemist from Lab Science Solution. And today I'm going to share to you on polarimeter technology in natural products, pharmaceutical, and other research applications. First of all, I would like to introduce on our organization. So we are a multi-vendor service provider established since 2005, and we are accredited by SAMM for wavelength. And we have also another branch in Singapore as well, and we are also accredited for wavelength under a Singapore Accreditation Council. And these are some of the services provided by our company, which includes installation of analytical equipment, training for analytical equipment, preventive maintenance, UV calibration, service contract, and so on. And under our organization, we also carry some of a number of brands for analytical solution. This includes Rudolph Research Analytical, which we are going to talk in detail later on for automatic polarimeter, Analytic ENA, Scala, and so on. We also have our own brand of UV spectrophotometer, which is known as Thomas Edison. So without further ado, let's go into our main topic today, which is on polarimeter from Rudolph Research Analytical. So as you can see here, Rudolph is a world leader in the manufacture and sale of high-quality reflectometers, polarimeter, saparimeter, and also densitymeter. And all these equipments are approved by International Commission of Uniform Method for Sugar Analysis. So over the years, Rudolph has invested heavily in Hello? Hello? I think someone turned on their mic. Okay, let's continue. Okay, so as a result, you can see we have produced products with automation where you don't have to introduce the sample manually to do the analysis. So there are a few series of automatic polarimeter, also known as Autopol, which ranges from Autopol 1 up to Autopol 6. And the Autopol that is available in i-CRIM is Autopol 6, and we are going to talk in more detail on Autopol 6 today. So there are a few functions that samples can be tested under automatic polarimeter, and it can measure optical rotation, specific rotation, concentration, and sugar degrees. So it comes with 6 wavelength, which is designed for the most demanding analytical and research laboratory. It also has a very high accuracy and reproducibility, and it comes with high precision patented temporal. So temporal is actually a built-in temperature control system where the samples can be tested from temperature ranging from 10 degrees Celsius up to 40 degrees Celsius. And it also has automatic wavelength accuracy check. Let's look into polarimetry. So polarimetry is the measure of chiral properties of optically active samples, or also known as enantiomers, without chemically modifying or destroying the sample. So, hello?
Sorry to interrupt. Yeah.
Yes?
We cannot see the slides.
Can't see the slides?
You do not share the slides.
Sorry. Sure. Can you all see the slides?
Yeah. Now can see the slides. All right.
I'm sorry. Okay. So the polarimeter is the instrument that we use. In this case, is automatic polarimeter, or also known as autopol 6, to make these measurements. So let's look at enantiomers. So enantiomers are optical isomers, or they are mirror images of a chiral molecule. So as you can see, the easiest example will be our hand. They are perfect image, mirror image of each other. However, you cannot superimpose them. So same goes to chiral molecules such as amino acids, as you can see here. It is a perfect mirror image of each other. However, when you try to superimpose it, it is not possible. There are a few components of polarimeter, which includes a light source, which can be a sodium lamp or tungsten halogen lamp, a polarizer, a sample cell, which is the tube which holds the sample, an analyzer, also known as second polarizer, and a scale to measure the rotation of plane polarized light. And the reading will be displayed in the display on the automatic polarimeter. And this is the simple schematic diagram of a polarimeter, which includes a lamp, polarizer. So when the light passes through the polarizer, it will vibrate in a single direction, and it will pass through the sample tube containing optically active sample. And the plane of polarized light will be rotated by the sample, and it will be analyzed by the analyzer, and the reading will be shown on the display. So according to wave theory, light is actually waves of various wavelengths vibrate in direction perpendicular to the direction of transmission. So as you can see, it is the wave that vibrates in many different directions. So when it passes through a polarizer, it only vibrates in a single direction, and it is known as plane polarized light. So the plane polarized light then passes through the sample tube containing optically active sample, and once the light hits the optically active sample in the sample tube, it will be rotated, and the rotated plane polarized light will be detected by the analyzer. So let's say if there's no any optically active sample present in the tube, so the instrument reading will be shown 0. This is because the axis of plane polarized light and the analyzer is exactly parallel. And if the sample is optically active, the plane polarized light will be rotated as it passes through the tube and when it hits the optically active molecules in the sample. Let's look at optical activity. So optical activity is the power of a substance to rotate the plane of polarized light. So in liquid and vapors, optical activity is a property of molecular structure. So enantiomers or mirror images will rotate the plane of polarization in exactly equal amounts, but in opposite direction. For example, let's say there are two mirror images, which is S2 bromobutane and R2 bromobutane. Both of them are enantiomers. For S2 bromobutane, the optical rotation will be positive 23.1. Meanwhile, for R2 bromobutane, the optical rotation will be negative 23.1. So as you can see here, The magnitude, which is the value, is exactly the same, except for the directions. One is positive and another is negative. So as I told you, rotation can be either positive, clockwise, or also known as dextrorotatory, or negative, anti-clockwise, or also known as levorotatory. So let's say when a sample has an equal mixture of enantiomers, the effect will cancel out each other, and this is known as racemic compounds. It is first detected by Louis Pasteur in tartaric acid. So the optical rotation are unique for a pure chiral molecule at a specific temperature and wavelength. So it is like our fingerprint. No two person can have the same fingerprint. Same goes to the sample, optically active sample. No two different optically active sample can have the same value. So here are some of the examples of specific optical rotation of molecule at 20 degrees Celsius under 589 nanometer. So as you can see here, this is the example that I've given to you just now. So it has the same magnitude. However, the direction of rotation is totally opposite. And another thing for you to note is when naming optical isomer, we have two ways of naming it. One is using D and L, which is dextrorotatory and levorotatory. And another way is using positive and negative. But we usually will be confused with D and L because for here we have an example of D erythrose. D under optical isomers means dextrorotatory, which is positive or clockwise rotation. However, when you see in D erythrose, it is not in the same case. So to prevent this confusion, we use positive and negative naming. So polarimetry scales are divided into two. In chemical and pharmaceutical industries, we use angular scale to calculate the specific rotation. Meanwhile, in sugar and food industry, we use international sugar scale, which the unit is degree Z, which indicates the percentage of sugar in 26 gram of sugar solution. And for sugar samples, usually it will be tested in a 200 mm cell. So as you can see, this is a formula under Bayes' law, where this is the specific rotation at a particular temperature and wavelength. And this is the measured optical rotation, which is obtained from the instrument. And L stands for tube length, which is the sample cell length, which ranges from 10 millimeter up to 200 millimeter. And C is the concentration of the sample, optically active sample, in gram per milliliter. So using this formula, we can calculate the purity or concentration of an optically active sample that is tested by automatic polarimeter. And temperature is one of the main factors which will affect the specific rotation of an optically active isomer. So in most of the applications, we will test the optically active sample at 20 or 25 degrees Celsius, and the temperature will be stabilized before the analysis. So as I mentioned in the earlier slide, we have a temporal technology, which is the temperature control program, which is available in the automatic polarimeter. So the temperature will be stabilized by the temporal technology before the analysis starts to take place. So this is the inside part of a polarimeter. So here, as you can see, there's a metal surface, which is a Peltier active surface, which controls the temperature. And this is the sample cell, which has a temporal passive surface. So when these two metal surface comes in contact, the temperature of the sample, which is filled inside this sample cell, will be altered. So as you can see here, there is a temperature probe which is inserted into the sample cell. So the sample temperature will be detected by the instrument as per set in the instrument. So the second main factor that will affect the specific rotation will be the wavelength. And as you can see here, there are few wavelengths that the optical samples can be tested under this polarimeter, which includes 365 nanometer, 405, 436, 546, 589, 633, and 880 nanometers. So at different wavelength, the specific optical rotation will be different as well. And as I told you before, length of the tube also affects the sensitivity of analysis. So the longer the tube length, the higher the sensitivity of the reading will be. So from this formula, we can easily calculate the concentration of the optically active sample in the sample cell. So this is the part where the sample tube will be altered according to the analysis. And the alpha is the measured optical rotation, and this is the value that we will obtain from the instrument after the analysis. And when the plane polarized light passes through the sample tube containing optically active sample, the plane polarized light will be rotated either clockwise or anti-clockwise, and it will be detected by the analyzer. So now let's look into the main part of our webinar, which is the polarimeter applications. So as we know, polarimeter actually can be used in various industry, which includes pharmaceutical, natural products, sugar industry, food industry, and so on. So in pharmaceutical industry, 56% of the drugs used are chiral molecule, and 88% of those who are marketed are usually racemates. So even though they have almost similar chemical structure, but their biological activities will be different. So the main reason why polarimeter is used in pharmaceutical industry is because one of the isomer will have more powerful effect in biological activity compared to the other. So for example, the lever rotatory isomer of beta blocker is has more active bioactive effect compared to the dextrorotatory isomer. So for example, propanolol is used to treat high blood pressure and also irregular heartbeats. Even though the S-propanolol and R-propanolol have almost the similar structure, however, the S-propanolol have a better effect, which is 100 times more active compared to the R isomer. And in another case, the S-citalopram is a tablet used for treatment for depression, but the S isomer having a better effect compared to the R isomer. So meanwhile, the methadone, which is used as a painkiller, the R methadone has a better effect, which is 25 to 50 times more analgesic effect compared to its S isomer. So this is why the pharmaceutical industry requires the products to be tested before it has been marketed to the public. The second major reason will be due to toxicology. Even though both of the enantiomers having the same, almost similar structure, however, the toxic effects can reside in one of the isomer, which can lead to death. So for example, in treatment for DOPA is a precursor of dopamine. It is used to treat Parkinson's disease. So however, only the one of the isomer is will provide a better effect compared to the other, where the D-dopa is more toxic compared to the L-dopa, which can cause the agranulocytosis. Agranulocytosis is a effect where white blood cell could not fight the infection in our body. So this is the reason, another reason why the pharmaceutical drugs should be tested, whether it's lever rotatory or dextrorotatory. So the Federal Drug Administration of USA requires manufacturers to identify and characterize the isomer of a racemic mixture. So in all over the world, all pharmaceutical industries will be regulated by a body. In Malaysia, it is regulated by NPRA, and globally, most of the analysis must comply and follow US Pharmacopoeia and European Pharmacopoeia. And as I mentioned in the earlier slide, we have a series of automatic polarimeter, which ranges from Autopol 1 up to Autopol 6. So for samples like ofloxacin, it has a specific rotation between +1 plus ±1 and 1% concentration. But using Autopol 1, it has a specific rotation error of ±2.0. Thus, Autopol 1 is not suitable for analysis of samples such as ofloxacin with a smaller optical rotation. When compared with Autopol 6, it has a better accuracy, which is 0.002, and a smaller specific rotation error, which is ±2. So this is more suitable compared to the Autopol 1 for the analysis. Meanwhile, for Autopol 6, it exceeds this requirement with ±0.0003 accuracy, which means it can test samples with even lower optical rotation. So another key factor in polarimeter testing is temperature accuracy. So under certain pharmacopoeia, they specified that the temperature should be within a specified temperature and temperature stability. So under US Pharmacopoeia, the temperature should be at 20 degrees Celsius. The temperature should not deviate more than ±0.5. And under British Pharmacopoeia, the temperature at analysis at 25 degrees should not exceed more than ±0.5 degrees Celsius. So in Autopol 6, it exceeds and meets this temperature requirement because the temperature deviation in Autopol 6 is just ±0.1 degrees Celsius. And there are many compounds or drugs produced used by pharmaceutical industry need to be included in a polarimeter as one of their quality testing under pharmaceutical industry, which includes steroids, vitamins, antibiotics, analgesics, and so on. So as we know, amino acid is a building block of proteins. So polarimeter is used to test amino acid for its purity and also concentration. It is not only involved in pharmaceutical, it is also involved in food industry as well. And pH will greatly affect the optical rotation of amino acids. This is because it has this carboxyl group and also amino group. And these two molecules are affected by pH, where it can be protonated or deprotonated. So it can also, pH also can affect the geometrical structure and also the optical rotation of amino acid. So some international pharmacopoeias requires the amino acids to be tested for its optical rotation at a defined acidity. So next, hello, yes. Okay, let's continue. Next, let's look into natural products. So the first natural products will be camphor oil. So as we know, camphor oil is used in ointments such as Vicks, which is a famous brand, and it has a strong aromatic odor. It can be produced by synthetically in lab, and also it is produced naturally by trees from the laurel family. However, to meet the supply and demand in the market, we cannot only depend on natural supply, so it has to be synthetically produced in the laboratory. So camphor, which is an optical isomer, it has both levorotatory and dextrorotatory form. However, the levorotatory camphor is more toxic compared to the dextrorotatory. Thus, camphor has to be tested, especially those which are synthesized by labs or produced synthetically, has to be tested before it has to be marketed or used in products. So as I mentioned earlier, the lever rotatory camphor has a higher toxic potential compared to the decamphor. So this is why the quality of camphor produced should be tested, and a polarimeter testing will produce an accurate result compared to other testing. So the second natural product that I'm going to talk about will be honey. So as we know, honey, there are two sources of honey that is produced. One is honeydew honey, which is produced by bees, and another source of honey will be nectar honeys. So nectar honeys are honeys secreted by plants, and both these honeys having totally different optical rotation, where honeydew honeys produce positive optical rotation, meanwhile nectar honeys produce negative optical rotation. So in this application, we can also detect whether there's a contamination between honeydew honeys and nectar honeys, and at the same time, we can also measure the concentration of sugar in the honey. So next will be starch. So starch is actually one of the main food supply that is consumed by humans, where starch will be broken down into glucose and provide energy in human metabolic activity. And it is also used in the paper industry to strengthen the paper and the surface of the paper. So starch actually can be tested through iodine tests or also enzymatic tests. However, polarimeter is a more accurate test to determine the quality of starch. Thus, polarimetry is used to test the starch sample compared to the iodine method and also enzyme method. So next will be sugar industry. So saccharimeter is an instrument which is used to test the concentration of sugar. In this case, as the polarimeter use the international sugar scale as defined by the i-CRIM, polarimeter can also be used to test the concentration of sugar in sugarcane juice, refined sucrose, saccharides, and other sugars. As I told you earlier, a saccharimeter is used to determine the purity of sugar. So in Malaysia, under Malayan Sugar Refinery, we have 3 Rudolph polarimeter in QA and QC lab to determine the quality and purity of sugar. And another advantage of using automatic autopol, Rudolph automatic polarimeter, is it can detect, it can test dark sample up to optical density of 4. And under food and beverage, compared to pharmaceutical, it is less stringent. So we do not test most of the food samples under polarimeter. Some of the samples that are tested under polarimeter will be monosodium glutamate, also known as MSG. We know it better as Ajinomoto, sugar, starch, milk, dairy products such as cheese, yogurt, and so on. So in milk industry or dairy products, we test lactose as a measure of sugar content. So lactose is actually considered as milk sugar. So it has to be controlled in food industry, especially in dairy products. Thus, polarimeter is used to test lactose in milk products and also products that is derived from milk, such as cheese, yogurt, and so on. So lactose is tested to ensure the quality in the lactose containing as well as lactose reduced dairy products. So in chemical research, analysis of optically active substance and structural analysis in inorganic ions in conjunction with the organically active substance such as bismuth, cadmium, copper, iron, and mercury in solutions are tested under polarimeter. In medicinal industry, usually sugar and albumin will be tested in urine, hormone determination, poison analysis, and so on. Next will be flavor and fragrance. So essential oil, as we know, all the essential oils are usually volatile. Automatic polarimeter under Rudolph has a temporal cell, and also it has a stopper which will prevent the sample from evaporation. So this will provide a better accuracy and a better results. So under research applications, polarimeter can be applied to test unknown samples, which is crystallized from various solvents or separated by HPLC. We can also use to characterize an unknown sample by obtaining the optical rotation from the instrument, and we compare with the specific rotation from the theoretical value found in the literature. From this, we can detect the unknown compound through polarimeter. Next, we can also use polarimeter to monitor changes in concentration of optically active component in reaction mixture, for example, as enzymatic scission or enzymatic cleavage. And we can also determine the configuration changes of salt macromolecules. And we can also investigate kinetic reaction by measuring optical rotation as a function of time. For example, we can test the concentration of sucrose due to enzymatic lysis over the time. And these are the applications that we can apply polarimeter in the industry and also research applications. So that's all from me. If you got any questions, you can ask me now. So shall we take a 5 minutes break before we start with our Q&A session?
Yeah, Mr. Naraj.
Hi, yes.
Yeah, sure. You can take a break. 5 minutes break.
Okay. All right. Thank you.
So we're going to have a 5 minutes break, and we'll come back after this for Q&A session.
Sure.
All right. If anyone has questions, please type it in the chat box. I will read it later during the Q&A session. Thank you.
I hear you. In session.
Any questions from the participants? For UKM staff, kindly fill up the link given in the chat box for registration in Sistem Urus Bakat. And also don't forget to fill in the feedback form. Link also given in the chat box. Thank you. I don't see any questions from the participants. So, Mr. Kanaraj?
Yes.
Yeah. Yeah.
Yes.
Okay, we have one question from Dr. Wan Aizat. Can we do all the application using i-CRIM polarimeter? And how do we properly prepare for the samples to be sent for the analysis?
Come again?
Okay, one question from the participants. Can we
Okay.
do all the application using i-CRIM polarimeter? And also, how do we properly prepare for the samples to be sent for the analysis?
Okay, under Autopol 6, which is available under i-CRIM, that is the most advanced automatic polarimeter. So most of the optically active samples in liquid form can be tested under automatic polarimeter. And the samples under automatic polarimeter 6, another advantage will be it can use a very little amount to test. So the sample should be sent in a airtight container, and then it can be tested. So before testing the samples, it has to be rinsed few times with distilled water to clean the sample cell. And we have to rinse with the sample as well a few times to make sure the sample is filled without any bubbles inside the sample cell prior to the analysis.
And another question, how pure should the samples be?
How pure? Actually, the polarimeter can be tested to, can test purity of a sample. So when the sample is sent, we must make sure the sample is clear without any suspended particles. So when we test the sample in the polarimeter, we can test for its concentration by referring to its specific rotation under standard condition. So as I've shown to you before in the Bayes' law, we can derive the formula, and then we will have that length, we have that observed rotation, which we will obtain from the polarimeter, and the specific rotation we can obtain from the theoretical value in the literature. And most of the times, the polarimeter, we cannot use polarimeter alone to detect an unknown compound, because polarimeter is complementary technology with other identification technologies such as MS, IR, and NMR.
Is there any minimum concentration to be detected by the machine, Mr. Naraj?
By Autopol 6, is it?
Yes.
So as I'm the minimum concentration. Hold on, yeah. The minimum concentration will be in 10 milligram. As Autopol 6 can detect as low as 10 milligram sample concentration.
Okay, Mr. Naraj, is there any more questions from the participants? I think Mr. Tanaraj already answered all the questions given in the chat box. I hope that the answer, the answer given answered the questions. Thank you, Mr. Tanaraj, for the informative presentation today. So I hope that all of the participants can switch on the camera. We start for the photography session. Okay, more participants want to switch on the camera? Right. 1, 2, 3. One more. 1, 2, 3. Okay, I think one page only. Okay, everyone, we have come to the end of our webinar today. I would like to thank the speaker and everyone for joining us today. We hope that this webinar has been beneficial and wishing the best for research you are currently embarking on. Stay safe and see you all in our future i-CRIM activity. I think we have CPMS webinar after, I'm not sure, poster shop. Okay. Yeah, we will be conducting a webinar on i-CRIM on 17 March 2022. So if you are interested in joining the webinar, kindly register before the 15 March 2022. Okay, everyone, that's all for today. Thank you again, Mr. Sanaraj and everyone. Take care and bye.
All right. Thank you.
Don't forget to fill in the feedback form.
Thank you, everyone.
See you all